We are happy to welcome you at the online lecture series from the NGS-CN Summer School 2024. As most of you already know, NGS-CN stands for the Next Generation Sequencing Competence Network. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We leave the link in the footnotes, and here is a QR code for your fast access. Welcome aboard, find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
Having me, and exactly, I am going to talk a little bit about sequencing applications. First, I'm going to talk about me a bit because just so you know who I am, I'm a project manager at the Competence Centre for Genomic Analysis in Kiel. I have been working there since 2019, and the CCGA is part of the sequencing network from the very beginning. If you ever want to visit us, you find us in this really very, very easy to find building because it's the only golden building without any corners on the whole campus. If you ask people for the amoeba, you will find us. If you don't want to find us personally, but online, you will find us on our homepage. That is also where you can make first contact, find out more about us, and everything else you need to know. And with that, let's take you one step outside of my office door as an introduction. And then you see this. This is a humongous poster that Illumina at some point in 2015 published. And to be completely honest, 2015, that is roughly 100 years in sequencing technology years. So this is so outdated, it is incredible. Still, if you have a closer look, what you see is that for every single DNA or RNA related application, there was a dedicated protocol already. I do not know half of them, to be completely honest, and I will not go through all of them because that would just take us 3 to 4 weeks, and we have half an hour. So we will dive into the few main ones, and to be completely honest, many of those dedicated protocols you see here, other technological solutions have been found since then. So let's start. As you have heard now quite a bit, the most relevant sequencing can be separated into DNA and RNA sequencing, while RNA sequencing is most of the time also DNA sequencing. I will talk about a few applications that are actually possible to do on both, on DNA or RNA, but most applications, general applications, are done on one specific molecule, DNA or RNA. So these are the things I will talk about. Some of them I will pretty much skip. For example, the expression level analysis, even though that's the most important one in RNA sequencing, but there is a whole lecture coming after that. So there is no point in going into detail now. But now let's dive in. We will start with microbiome sequencing. So as we heard in the questions before, there are a few people doing microbiome sequencing in the audience. So for you, it's probably kind of obvious, but what you see here is an overview of several different body tissues of the human body, and an overview which bacteria, in this case only bacteria, are represented in which part, which ones are dominant, which ones are not there, etc. So there is a profile, a microbiological profile to pretty much any site in the body, or basically any place you can swap or dig or extract, you will find a microbiome there. So how can you access the microbiome? So your questions in microbiome analysis is basically who is there and what are they doing? And you can start with, Alex already mentioned that, with sequencing barcoding regions with 16S and 18S, you will access bacteria and archaea. With ITS, you will access fungi. You can also sequence COX, which is not necessarily microbiome sequencing, but it can, for example, give you information on what kind of prey a predator ate, because that will unlock animal species. It's a PCR-based method. It relies on the fact that all over the tree of life you have highly conserved genes. 16S is a ribosomal subunit. And these highly conserved genes you can have the possibility to design primers in sequences that are super conserved, and in between you have variable regions that can be used to identify a species or at least a genus. This is possible for short and long reads. For long reads, you just have more PCR product, which means you have a higher chance of classifying it to a lower level, so sometimes even strain level. The similarity to a reference database will give you the information on which genus or species you detected. The positive sides are it is cheap. There are very many homebrew protocols out there. It's basically PCRs, and then you can sequence. It is very well established. It has been done for pretty much every clade on the tree of life. I assume, I haven't checked, but if you can design a primer, you can also sequence it. The cons are definitely that they are only as good as the reference database you are using later on in your analysis. So you have to think about if you are working with a niche that has been explored before. So if you are working with any part of the human body, you are probably relatively fine. If you are working with some bacteria out of a volcanic crater, you probably are not that fine, to be honest, because that's very special and you probably will not have a lot of representation. So you can probably not identify your species of interest. Also, your resolution and outcome will be only as good as the primers are. So you can go to the next level, that is metagenomics. Alex also mentioned that before. That means it will sequence all DNA that is present in your sample. That means viral, fungal, bacterial, archaeal, animal, plant. And that has been isolated. That's why Alex mentioned that you have to optimize your isolation protocol. If you want to see viral and bacterial and plant DNA, you will have to work on getting all of them open equally. The pro is that it represents all DNA within the sample, and you even have the possibility to assemble a genome if you are lucky. The cons is that it's obviously more expensive because you are sequencing more DNA. It is also more computationally expensive, while you can do some 16S analysis even on your laptop. You cannot do that with metagenomic analysis. It also shows you environmental DNA. That means if you are sampling a soil sample and 3 weeks ago a deer pooped there, then you will sequence dead deer poop microbiome and not soil microbiome. And to be focusing a little bit more on what is actually active, you can sequence metatranscriptomics, meaning you isolate the RNA from your sample. And that way you can find only pathways that are active and used. You can also use it to determine who is there, but this sometimes the function is a lot more informative than the identity, talking about volcanobacteria again, where you can see what they do without necessarily having to assign a species to it. The cons are absolutely the RNA quality. Depending on where you sampled and what you are aiming for, the RNA extraction methods are quite harsh. So it is sometimes not ideal to get perfect quality. The second one is that rRNAs, so ribosomal RNAs, are very overabundant. If you are not working in human, the depletion of rRNAs is anyway not very efficient. So you will have a lot of rRNAs in there. Talking about hosts like human, Alex already mentioned it. I just want to repeat it. It is better to get rid of the host during sampling and lab than later on do it during the bioinformatic analysis. And this here is expensive as well as the metagenome. And with that, we go away from the microbiome up to a certain degree and go to whole genome sequencing. The question here is there a reason on genome level for what I see? Of course, you have a disease, you want to know if that has a genetic origin. So you have several different types of reference of genomes. To actually work on genetic data, you ideally have a reference or a reference genome that you can create with long reads and with short reads. For long reads, it is the absolute gold standard if you combine it with Hi-C, which gives you extremely long range distance information, or with optical mapping that Max mentioned briefly. Phasing is possible, so you will have haplotype phased a genome, and with long reads, the methylation pattern is immediately included, which is also quite nice to have. The disadvantage, as Max already mentioned, it is very expensive, and you need very high DNA quality and DNA quantity, like that. For short reads, the start is relatively cheap compared to long reads. The threshold to get started is also a lot lower because the DNA requirements are a lot lower. The disadvantage is that your genome will not very continuous. You will have a lot of fragments. There is no phasing possible. And to get to the one reference genome, we are all that has kicked off the sequencing era, I would say, the human genome. It has been published in 2001 in Nature. And as Max already mentioned, it was very expensive. It took a lot of time, and it was a Sanger genome. So I would say medium long reads. And even though it was considered finished, it had a lot of gaps. Now, at 21 years, and a lot of research done in between, but finally in 2022, the first telomere to telomere chromosome resolved genome was published in Science. What I want to point out, there has been 21 years on the human genome without needing a perfect genome. So what you have to think about is, what do I need for my purpose? How good does it have to be? So there is quite a range of how good a reference genome has to be. You usually will use your reference genome for resequencing purposes. So to analyze population structures, do a mutation screen, do genotyping by sequencing. And this you will mostly do with short reads. So you need less coverage, meaning it is relatively cheap again. You will get up to a certain degree intronic and exonic information. The disadvantage is that it is, for large screening projects, it is only medium throughput, I would say, depending on your budget. You will need a reference genome, and a relatively large region of the genome is actually inaccessible for short reads. So be it either because there are structures that you can't resolve bioinformatically, or there are large repeat regions that are just not possible to access by PCR amplification that you do in short read sequencing. So either you go for long reads, which is a lot more expensive, or you have a rough idea where you want to look for the data you have and switch to targeted sequencing. So the targeted sequencing basically is, your approach is, there is a genomic background of what I'm looking for. I suspect, hope, or know that it is somewhere in the coding region. Max already mentioned it. The DNA is structured into introns and exons and a lot of other DNA regions. If you are sequencing the exome, you are sequencing only the coding regions, so the exons, which comprises roughly a slice of the apple of around 1%. You can whole exome sequencing, you would do bait-based. So that means that you have DNA, or in some cases RNA baits, bind your DNA, enrich the bound DNA, and sequence that one. There are several different methods to do so. If you want to go into detail, that really depends on the kit you are using. It is a lot cheaper than whole genome sequencing because you only need 1% of the data. It will cover most coding regions. It is a lot less problematic if you have low quality DNA like cell-free DNA and FFPE DNA. The throughput is considerably higher because it is so much cheaper, and relatively little previous knowledge is necessary. You just rely on that it will be in the coding sequence, and this is also the biggest disadvantage. You will find only what is in the coding sequence and in the previous years, it has become more and more apparent that not all underlying causes of disease are in the coding sequence, but a lot of it is in the non-coding areas, which you will never find if you are doing exome sequencing. If you know a lot more about where you want to look for your region of interest, like a gene family or something like that, then you can go for a custom panel or an off-the-shelf panel. There are plenty of them for like the pan-cancer panel. It is a lot cheaper, again, depending a little bit on the panel size and what you are looking for. It is in considerably higher throughput because of that. However, if you are planning a custom panel, the threshold for optimization and getting it to run is relatively high. The initial costs are relatively high. So you only should do that if you are indeed having a large sample number to screen. And of course, you need to have previous knowledge to know exactly where in the genome to look for. And getting it even cheaper and even narrower is a PCR-based approach. That is very similar to 16S sequencing. It is very cheap. It is in considerably higher throughput because of that, but it is limited to very small subsets of genes. To establish a protocol with multiplex PCRs might be very time-consuming because you have to work hard on specificity and sensitivity. It is limited to relatively easy to amplify genes, and it is limited to genes that you can have a proper primer design and still identify what you are looking for. You can actually do all those approaches with short and long reads. They are mostly done with short reads, though. And with that, we are switching gears a little bit and sequencing something that has nothing to do with the direct sequence as such, but you are looking at what is on the DNA, what does the DNA look, or even proteins, because you can actually sequence proteins with NGS, sort of. That is usually called feature sequencing or structure sequencing. And one of the first things I want to tell you about is methylation. With short reads, there are two possibilities. You can do a restriction-based approach to enrich for methylated sites, or you can do whole genome sequencing, which of course is more expensive. The principle behind methylation sequencing is always that the unprotected, so unmethylated Cs in your sequence will be converted into uracils, which then will be sequenced as a T. So if you are having a reference genome, you compare your methyl-seq experiment 2 everywhere where you have a C and read a C in the sequencing that has been methylated, everywhere where you have a U or a T instead of a C that is an unmethylated C. You can do that either with bisulfite treatment, which is very harsh and restricts you to a shorter read length, or with an enzymatic method, which is actually giving you the pretty much same result. It just gives you the possibility to sequence a little bit longer reads. If you are sequencing long reads, then you have the big advantage that you have a relatively unbiased approach. It is possible with PacBio and Oxford Nanopore, native methylation is detected, which also means that you can detect C methylation and A methylation, which is mostly bacterial. It is a lot more expensive, but what you do not need is a reference genome, unless you want to compare it to something, because if you only want to detect methylation status, you already have your reference genome in the same experiment. Let's switch to chromatin status. So as we know, DNA is packed, and then there are several possibilities to access the chromatin status by cross-linking the DNA with the histones. and then digesting away what is not, or depleting what is not of interest. So either you are sequencing the accessible chromosome, ATAC, or the bound chromosome and bound part of the chromosome and deplete the rest. And with that, you get long distance information. That's why you can use Hi-C, for example, also for scaffolding. And with that, we switch gears again quite a little bit, because now we are talking about proteins. First of all, protein interaction sequencing, like chip sequencing. There you sequence protein-bound DNA, which means that you enrich by using an antibody against your protein. You enrich bound DNA, digest everything away that is not bound, and sequence what is the DNA that is left. And that works very well for transcription factors and so on. Or you can actually sequence protein expression patterns. The highest throughput at the moment I know of is Olink, where you have a panel of more than 3,000 proteins that you can tag. The principle is shown here. You tag, you bind your protein with, in that case, 2 antibodies. Those antibodies are linked to a DNA sequence of a certain length. And by sequencing that DNA sequence, you can identify which protein has been bound. With that, you can have comparative analysis of protein expression patterns, which I think is pretty cool. You can do that on a little bit of a larger scale with CytSeq, for example, and this is used a lot in single-cell sequencing and spatial sequencing. So in single-cell sequencing, what you do is you have a rather heterogeneous cell type, and you assume that your different cells in your sample have a very different expression pattern, chromatin state, so like attack, you can do attack, mutation state in tumors, or protein signatures. Then you have to employ CytSeq and enrich protein-bound cells, cells that express a certain protein, that way around. What you do is, this is the 10x principle, just because they are the leader on the market. What you have is here, it's gel beads that contain a certain barcode. You add your cells, and in theory, one cell binds to one of those gel beads, then is encapsulated in a droplet. And within that droplet, the whole cDNA synthesis, including tagging with that barcode, happens. So at the end, you can retrace your steps, which expression profile is happening in which cell origin in your sample. There are plenty of other technologies, for example, the BD Rhapsody, which does the same thing, just plate-based. There are methods with lower throughput. If you just don't have that many cells, it's not necessary to do such an experiment, or methods where you do not need an extra device, like from Singleron or from Parse Biotechnologies. The big advantage of all of them is high throughput. You can access roughly 10,000 of cells and more. You get information on the expression state in single cell resolution, and with most technologies, you can also combine it with expressed proteins or with TCR and BCR profiling, for example. The disadvantages is really that it is very expensive, and if you are working in a not very well established organism, then it is kind of hard to identify which cell types you have in there, because you have to annotate the cell type based on the expression profile. And this is hard to do if there is not a broad database. And one thing that you have to have a look at, not all cell types are equally accessible with each and every method. Some cell types work better with 10x, some work better with BD, etc. So you have to have a look at that before you even start. If it is really important for you to know what does the neighboring cell of my cell of interest do to change the expression pattern, you might want to go into spatial transcriptomics, which is pretty nice. because it gives you expression patterns combined with where in your tissue this expression pattern occurs. Basically, the big advantage is it combines histology with expression levels of transcripts. So what you have, it is a similar principle like single cell, just that your barcodes that label your transcripts to trace back, in that case, the location of the cell origin, are spotted onto a microscopy slide. So what you do is you put your histology sample on one of those 10x Visium, in that case, slides, do your normal histology, then release the RNA from your sample and prepare a library that will tell you at the end exactly where your transcript has been expressed. The level of expertise needed here is extremely high because you need someone who is good in histology and is good in molecular biology. It is very, very expensive, and the resolution is depending on the size of your cells, not one expression profile, one cell. So you might actually have mixed expression profiles. However, the method is still super valuable and really, really helps a lot understanding tissue interactions and dynamics. And with that, we are switching to bulk RNA. As I said, I will not talk about that in detail. It is probably one of the most used methods. That's why it deserves an extra talk. You probably all have seen these kinds of graphs in papers where you see fold changes, overlapping genes, and clustering of samples based on their transcription profile. It is a lot cheaper than single cell. It is very well established. The really important things are that you pay attention to RNA quality and that your experiment has statistical power. And with that, I will switch to something a little bit more specialized, isoform sequencing, which is taking into account that you have alternative splicing. So one mRNA or one coding sequence, one RNA will not give rise to only one protein, but it can give rise to several proteins. And as you can see in this example, they can be vastly different. Why is that an issue? Because with short read sequencing, you have to find a read that actually spans your splice site to be sure which isoform is expressed. And when you have an isoform that is identical until here, but different in the end, then you cannot say if you have that isoform or that isoform, because you do not have a link from the first short read spanning the splice site to the second short read spanning the splice site. If you do long read sequencing, that is pretty simple because you will just basically sequence from the beginning of your transcript to the very end of your transcript. So there is no ambiguity about which isoform you actually detected. There is cDNA-based methods, very robust. You enrich for transcripts because you do a reverse transcription and enrich usually via a poly-A tail or a workaround for bacteria for transcripts. The disadvantage is that it introduces some bias towards transcripts that are accessible to reverse transcription. So as Max already mentioned, there is direct RNA sequencing in Oxford Nanopore. The big advantage is it does not only sequence mRNAs, it sequences all RNA species. I come back to that in a second. There is no PCR or reverse transcription associated. So you can see all modifications directly. There is the secondary structure is not a problem. The big disadvantage is when you are interested in only transcripts, it is not only mRNAs you are sequencing, but all RNAs, unless you introduce again a biased step of depleting certain RNA species or enriching for certain RNA species. And because I mentioned now accessible to reverse transcription and secondary structure, I will just go for a second to non-coding RNAs, because many people are interested in non-coding RNAs because they regulate quite a lot of processes in any kind of organism. However, they are very difficult to find in your sequencing data. For one, they have secondary structures. If you are going for short-read sequencing or cDNA-based sequencing, very often not the whole secondary structure can be transcribed into a cDNA. So you will have, sometimes you will lose hairpins and stuff like that. Then you have circular RNAs. They are obviously not accessible at all to cDNA transcription. And you have microRNAs. In that case, it's a pre-microRNA, but that are super short. So what I'm saying is that if you are interested in non-coding RNAs, you have to read up a lot into very, very, very dedicated and specialized protocols usually, because you cannot get them with standard protocols. When you do native RNA sequencing with Oxford Nanopore, the secondary structure is not that big of a deal because your motor protein acts as a unwinds the secondary structure. So you can actually have relatively easy access to, for example, tRNAs if you are interested in those. And with that, I thank you that you joined for that a little bit wild ride, because it was a short time for a lot of topic. Just to introduce you briefly to the people doing the work. Again, here is our homepage. You can follow us on LinkedIn, and this is where you find us in person. And now I'm happy to take your questions.